On small farms across Europe in the late 19th century, you could spot a strange detail if you looked closely. A thin strand of copper wire wrapped around a plant stem. No machines, no chemicals, just metal pressed against living tissue. The farmer would wind it tight with calloused fingers and walk away without explanation. You are told plants need soil, water, light, and air. That is true, yet farmers have always experimented beyond the textbook. Copper caught their attention because it did not rust like iron. It held its shape. It carried an electrical charge. In damp soil, it reacted in ways that felt almost alive. Copper has been used in agriculture for centuries. Vineyards in France relied on copper sulfate mixtures to fight mildew. Orchard keepers hammered copper nails into trunks to slow fungal rot. These practices were recorded in farm journals and local notebooks. The wire trick grew from the same thinking. If copper can protect a plant, can it also stimulate it? The method was simple. A short length of bare copper wire, about the thickness of a matchstick, stripped clean, no coating. The farmer would pierce the lower stem of a tomato or pepper plant. The wire slid through the green flesh just above the soil line. Sap beaded at the wound. The metal stayed in place. The metal. In some regions, the wire was not pushed through. Instead, it was wrapped tightly around the stem. Just one full turn, pulled snug. The pressure would bite into the outer layer, but not enough to cut the plant in half, just enough to restrict the flow. You could even hear sometimes the faint creak of fibers under tension. This was not superstition alone. You see, plants move nutrients through tiny internal tubes. Water rises from the roots, and sugars travel down from the leaves. When you partially restrict that downward flow, the plant responds. It thickens the stem, redirects energy, and accelerates certain growth processes. The stress triggers defense chemistry. Copper itself plays a biological role. It is, in fact, a micronutrient. Plants require it in small amounts for enzyme activity. It assists in photosynthesis and respiration. Too much will poison the plant, but too little will weaken it. The wire introduced trace contact at a fixed point. In damp conditions, small amounts of copper ions could migrate into the tissue. You can actually test the physical effect without involving any chemistry at all. Just take a soft garden hose, pinch it slightly, and you'll notice the pressure behind your fingers starts to build up. When you release it, the flow surges forward. A plant stem, interestingly enough, behaves in a similar way. The restriction changes the internal pressure, and you often see that the cells above the wire swell up, becoming thicker and sturdier. During the interwar years, some growers in Eastern Europe claimed they were seeing higher yields from tomato plants that had been treated this way. They reported heavier fruit clusters and a shorter time to ripening, which is quite remarkable. These accounts mostly appeared in local farming bulletins rather than in academic journals. The photographs, though, showed stems with dark metal bands glinting in the sun. Step into a humid greenhouse for a moment. You can smell the sharp scent of soil and leaf mold, and condensation drips from the glass panes above. In one row, the stems are smooth and untouched. In the next, thin copper strands catch the light. The treated stems look just a bit thicker above the wire, and the fruit sets lower and denser along the vine. Skepticism followed, of course. Agricultural scientists, well, they demanded controlled trials. Some experiments showed modest differences, but others showed none at all. Results, honestly, depended on soil composition, plant variety, and climate. The trick never became standard practice. It just sort of remained on the margins, passed by word of mouth. The wound itself matters, you know. When you pierce a stem, the plant actually activates its repair mechanisms. Lignin strengthens the damaged area and defensive compounds increase. Certain stresses can even increase fruiting as the plant senses a threat. It shifts from leaf growth to reproduction. Survival first, seeds before death. Copper has antimicrobial properties, that's for sure. It disrupts fungal spores and bacterial cells. In wet seasons, stem infections can enter through tiny cracks. A copper contact point may, well, reduce local infection risk. Farmers who noticed fewer rotting stems credited the wire. They saw results in the field, not in a laboratory. There is also the question of bioelectricity. Plants generate small electrical potentials as ions move across membranes. Copper conducts electricity. A wire inserted through moist tissue creates a pathway with different electrical properties than plant cells alone. The scale is microscopic, yet plants are sensitive to subtle signals. Hold a length of copper in your hand. It feels cool and firm. Bend it, and it resists before yielding. Press it against a living stem, and you sense the contrast between rigid metal and flexible fiber. That contrast is the heart of the method. Stress applied with precision. Survival gardeners during shortages experimented with any edge they could find. During the Second World War, kitchen plots fed families under rationing. Gardeners shared tricks in quiet conversations over fences. A twist of copper cost little. If it increased yield even slightly, it was worth the risk. Not every crop responds the same way, you know. Thick woody stems tolerate piercing better than, say, delicate herbs. Tomatoes, peppers and aubergines were common candidates, but soft lettuce would just collapse under the same treatment. Understanding plant structure is absolutely essential. A hollow stem behaves quite differently from a solid one. If you attempt this, you must control variables. Use clean, bare copper. Insert or wrap just once. Do not repeat along the same stem. It's important to monitor for wilting or darkening tissue. Excess stress kills, unfortunately. The goal here is stimulation, not strangulation. Precision over force every time. Picture a single tomato plant in open soil. Sunlight warms the leaves. The earth smells metallic after rain. A thin copper strand crosses the stem just above the ground. Weeks pass. The plant sets fruit. You weigh the harvest against an untreated neighbor. The scale gives you your answer. Modern agronomy focuses on soil chemistry, genetics, and irrigation systems. The copper wire trick sits outside mainstream practice because, well, evidence remains inconsistent. That does not erase the historical record, though. Farmers observed patterns, they experimented, they adapted to their environment with what they had. You are not chasing magic. You are studying plant stress response. Controlled injury can alter growth. Micronutrient contact can influence enzyme systems. Antimicrobial surfaces can reduce infection points. Each factor alone is small, but combined they may shift outcomes in specific conditions. There is risk. Piercing creates an entry site for pathogens if hygiene is poor. High copper exposure can accumulate in soil over time. Responsible use demands restraint. One plant first. Careful observation. Written notes. Compare results season by season. Serious growers respect evidence. If the treated plant outperforms the control across multiple cycles, you record it. If not, you discard the method. Survival skill means, well, testing claims under real conditions, not repeating them blindly. The image of a copper wire around a green stem endures because it represents self-reliance, a simple material, a deliberate act, a direct intervention in natural growth, no dependence on complex supply chains, just knowledge and nerve. Stand in your garden at dusk, leaves rustle in a light wind, soil cools under your boots. You decide whether to experiment or not. The plant will respond according to biology, not belief. Your task is to observe without bias. The lasting lesson is this: stress shapes growth. Controlled pressure can, redirect energy. Whether through pruning, grafting, or a strand of copper, the principle remains. Understand the mechanism before you apply the method. Test carefully. Document everything. Share verified results with others who value accuracy over rumor. If you value forgotten practices grounded in history and tested by your own hand, subscribe to Forgotten Fortitude and share this with someone who still believes experimentation matters. Carry the knowledge forward and, well, let the harvest speak for itself.